All right, so picking up with the protein information, once you have, the primary, secondary, tertiary, and maybe even quaternary structure, um, sometimes we have to, especially when it's just a single polypeptide chain forming a protein, um, we used to think that this process just happened kind of on on its own, right? Spontaneous. It was just kind of innate, this folding. But they since have discovered proteins called chaperone proteins that actually help with that folding process. In terms of what does it do, we believe it maybe provides the correct microenvironment for protein folding, for the most optimal conditions. If there are problems with chaperone proteins, we have even identified diseases caused by not the actual protein, but the chaperone protein that creates a problem for the protein um, folding, like the cystic fibrosis. So, in some cases, in some types of cystic fibrosis, the protein that forms the calcium channel is actually correct. It has the right amino acid sequence, but it doesn't fold correctly, which could be a problem due to chaperone protein. So here's just a diagram. It kind of looks like a crock pot, sort of, right? The chaperone protein, it loads the protein, the polypeptide. It goes in, it creates, again, some kind of correct environment with an input of energy, and then out comes the correctly folded protein. Now, protein's shape is very specific for the job it does. Why? Because shape is important to molecules, because how do proteins work? They work by like a lock and key fit, especially like with active sites and substrates in an enzyme. So that structure is really important. If something occurs to the environment of the protein, it can actually denature the protein. So if there is a change in pH, if there is a change in temperature, perhaps if there's a stronger concentration of ions in a solution, that can cause problems for the protein, and that protein can unravel and create an unfolded protein, and we call that denaturation. Denaturation, that sounds like a strange word, but it denatures. The protein denatures and breaks down, right? So properly folded and then denaturing, the whole thing kind of falls apart. Now, that takes us to our last category of macromolecules. Lipids are characteristically known by one major fact, that they are insoluble in water. And that is due to all the nonpolar carbon-hydrogen bonds that make up lipids. They are mainly hydrocarbons with just a very little amount of oxygen in them. But fats, oils, waxes, some fat-soluble vitamins are in the category of lipids. So fats in general, types of fats, we call them, there are triglycerides. Triglycerides are a commonly measured type of fat in the human body. We take a blood sample, we check the triglyceride level. It's one glycerol and then 3 fatty acid chains hanging off of it. The fatty acids are what create the lipid nature of these triglycerides. So fatty acids are just long chains of hydrocarbons within one single triglyceride. Those chains don't have to be identical. The length can vary, and what also varies with these fatty acids is whether or not they are saturated. If they are saturated, that means that there are no double bonds between the carbons. Every location is full of or carries a hydrogen on it, as opposed to a double bond, carbon-carbon double bond. So saturated fats are those fats that stay solid longer in our bloodstream because they have a higher melting point, and they tend to be from animals. Unsaturated fats tend to have one or more double bonds, which causes a kink in the chain, which allows them to spread out, allows those fatty acids not to be packed on top of each other. If the chain is kinked, right, it's going to kind of spread things out more. Unsaturated fats, they tend to stay liquid in our bloodstream or in our bodies. They have a lower melting point, and they typically come from plants. You've heard of trans fats. Trans fats are artificial fats produced in industry. They're used to kind of as a food preservative and to make certain types of foods, taste better, have a better consistency, but ultimately probably the most unhealthiest fat that you could eat. So here are two types of triglycerides. Here is a triglyceride with 3 fatty acid tails that are saturated, and then here is a triglyceride that has 3 fatty acid tails that are unsaturated. So you can see the kinks in the tails, and those kinks cause them to spread out more, and as things spread out more, they tend to be less solid, more liquid, and in this case, liquid at room temperature, where the saturated is solid at room temp. Another category of lipids are phospholipids. We talked about these when you were freshmen quite a bit in membrane structure, and that's their major job. They are made up of a glycerol with 2 fatty acid tails, right? We called those nonpolar tails. We called the glycerol molecule the head of the phospholipid, but it's a phospholipid because there is a phosphate group attached, and that makes phospholipids have kind of an amphipathic personality. They have dual personality. So the polar head has a charge, and so it's attracted to water, but the nonpolar tails are hydrophobic. And these are the major source of all biological membranes. So 4 different models here, right? This is how we always drew it in bio. But here's your polar phosphate head. It's made up of a glycerol molecule with a phosphate group attached. We don't really talk too much about the choline there, but the phosphate group is what makes it a polar end. And then the tails, here you have one saturated, maybe a slightly unsaturated, one saturated tail here and one unsaturated tail there. Then there's also micelles, which are lipid molecules that actually have a polar head and nonpolar tails. And they just kind of form and coalesce into little droplets. There is theories out there about their origins and how maybe these were the origins of cell membranes, but micelle then is just a cluster of lipid molecules that orient the nonpolar stuff away from water and the polar parts towards it. So when you take phospholipids and you create this more complicated structure, especially in living organisms, you have that double layer, right? The bilayer. So the phospholipid bilayer is indicative of living organisms made up of cells, and this creates cellular structure, right? It makes the membrane, it creates all the membranes of organelles, and again, the hydrophilic ends point towards the water, both outside the cell and inside the cell, and then the tails point inwards.